So today one of the first things I wanted to do was review a little bit about osmosis and diffusion, because that is the purpose of this lab. And the two experiments we would be doing today if we had the full lab time focus on osmosis and diffusion. And diffusion, as we know, is if you take some substance and stick it in a solvent of some sort. Let's just take our strawberry flavored sugar cube here, right? So we take a strawberry flavored sugar cube and we drop it in a bank of water, and we have a very high concentration in this little sugar cube, right? But what's going to happen to it after a while? It'll diffuse, exactly. So these things, these molecules of sugar, they'll start to dissociate and then they'll start bumping into one another. And so they have a high probability of collisions with one another. There's a certain amount of energy in this substance here, and there's a high probability that these sugar molecules will bump into one another, and they'll keep bumping into one another until they are evenly distributed and dispersed throughout the liquid. And so now what we have is a solution, which is a uniform mixture of two or more substances. And so the stuff that is dissolved is the solute, and the stuff that's doing the dissolving, in this case water, is the solvent. And so the solution is a uniform mixture of two or more substances. So if you let this sugar dissolve and you wait long enough, then all those molecules will have bumped into each other until they've achieved the most space that they can. Make sense? So it's kind of like if we had 20 students in here, or let's say we had 60 students in here, and then we opened both doors. Probably a lot of them would want to go out because it would be really tight in here. And the same is true with the sugar molecules. When they're in the sugar cube, it's really tight in there, so they want to create as much space between them as they can. Now they'll continue having little collisions with one another, but the collisions will not create any more space. They've already sort of created the maximal space. So there's movement, but there's no net movement. Does that make sense? So they're still bumping into each other, but very much less often than they were when they were in a concentrated cube. And so as they're bumping around and they have this random motion, that's just going to continue. And of course that will be dependent on lots of different factors, like how hot this solution is and so forth. But one of the definitions of diffusion is things moving down their concentration gradients. That is, moving from a high concentration to a lower concentration until everything's equalized, right? So that is diffusion. All right, next what we're going to do is we're going to talk about osmosis, which is a special case of diffusion. So your book makes an example of a U-shaped tube. And we're going to talk about the diffusion of water through a selectively permeable membrane. And that is really the definition of osmosis, is the diffusion of water through a selectively permeable membrane. So when you think of a cell, you remember the cell is a phospholipid bilayer, right? So the plasma membrane is a phospholipid bilayer. It's mostly lipid, right? So water can't just diffuse through lipid, and it can't move through lipid, but cells have these little channels in them, these little protein channels, called aquaporins, which literally means water channel, right? So the cells have little holes in them, specifically for the passage of water. And for this reason, we call them selectively or semi-permeable membranes, because they're permeable to water, but not necessarily to other things. So if we have a membrane here that separates this tube, and it has little holes in it or little pores in it that are just big enough for water to pass through, but not big enough for things like sugars or starches or salts to pass through, then we could have solutions in these tubes, or on either side of these tubes, that have different concentrations of solute in them. So I'm going to label this A and this B. And if we had, for example, pure water over here, and then we filled this side up with an equal volume of fluid, but we had some solute in it, and the solute is too big to pass through this membrane, right? So the solute is too big to pass through the membrane, but the water can. Now the water wants to do exactly what anything else does when it diffuses. It wants to go down its concentration gradient from an area of higher concentration to an area of lower concentration. And of course, this is what we call osmosis. It's just for water. So if we have a bunch of water molecules on this side, but on this side we have the same volume of fluid, but some of that volume is being taken up by the solute, then which side has the higher concentration of water, A or B? A, exactly. So where will water want to go? A will want to go towards B because there's less concentration of water on the B side. So if we wait and we just fill these things up like this, and remember, these pink particles can't get through the membrane, but the water can. The water is going to go toward the other side. And after a while, the level of A will go down and the level of B will rise. Simply because water basically is trying to equalize itself on both sides. So if we had two separate concentrations, and it doesn't matter really what they are, but let's just say we started with equal fluid volumes on both sides, and we had two different concentrations of solute in solution. So let's say we had a 10% sugar solution on this side and a 40% sugar solution on this side, right? Water's still going to move to the 40% side because there's less water on the B side than there is on the A side. But if we let it sit long enough, basically what happens is the water level is going to go down. The solute particles, the number of solute particles on either side is going to be the same, but now the proportion of water to solute will be evenly balanced. So by the time this thing finishes, these concentrations will be equal. Does that make sense? So that the water has moved towards the solute. We say water moves to solute simply because it's going to try to even itself out proportionally with the amount of solute, right? Does that make sense? So this brings about another important definition. And let's go to a cell now. So let's take an example of a cell. So we'll use a red blood cell, because a red blood cell is a pretty good example. So here's our sort of almost donut-shaped red blood cell. And most cells have some solute in them. So they're going to have some particles, some salt, some proteins, stuff like that. So here's our red blood cell. And if we put this thing in distilled water, then the distilled water has less solute in it than does the red blood cell, right? So now we can compare the two solutions. So if you have two separate compartments, in this case we have the cell versus what's outside, the extracellular fluid, we have two fluid compartments. The cellular one is going to have more solute inside of it. So it's called hypertonic. Anytime we have more solute inside, or I should say more solute in one solution compared to another, it is hypertonic. The solution, which actually has no solute in it, on the outside is hypotonic. So we compare two solutions to one another. The one with the more solute is hypertonic. The one with the less solute is hypotonic. And fluid will move from the hypotonic solution to the hypertonic solution. So we got little passageways in the cell for water. So where's water going to go? Inside the cell. So the cell will swell. In fact, if you put it in a really hypotonic solution, it may swell so much that it explodes, right? That it lyses. So in this case, the way I like to remember it is if you put a cell in hypotonic solution, it could explode. So an easy way to remember hypo, explode. Now, if we reverse the situation now, and we had a lot more solute outside the cell than inside the cell, let's just say we put it in a really, really salty solution, right? So we got a lot of solute outside, a lot more solute outside compared to inside. So now where's the water going to go? It's going to go outside, exactly. Because now the solution outside is hypertonic because it has more solute, and the solution inside is hypotonic So what's the cell going to do? It's going to shrink. It's going to shrivel up. And in fact, this will continue. The cell will kind of shrivel up here. And it will continue until the fluid concentrations are balanced on both sides. So our cell will get kind of shrunken. And then by the end of it, the solutions should be isotonic, right? That means you have the same amount of solute and solvent, so the solution concentrations are the same on both sides. Make sense? So that's just basically how diffusion and osmosis work. So diffusion is any molecule moving down its concentration gradient or substance. Water is a special case of diffusion that we call osmosis. And that is water moving down its concentration gradient through a semi-permeable or selectively permeable membrane. Now, who watched the potato core experiment? Okay, so everybody knows we took some potato cores and we basically took a cork bore and we cut out some strips of potatoes. And potatoes have a fair amount of solute in them. They've got a fair amount of starch in them, right? And so what did we do? We cut these things. In the video, I think they cut them all equally to 6 centimeters, right? And then we put them in different solutions. We put them in a distilled water, pure water, and then we added salt to get 5 different concentrations of salt solution. We had a 1 gram in 100 milliliters, all the way up through 5 grams in 100 milliliters. So by the time you got to the 5 grams, you got a super hypertonic solution, right? And typically the distilled water is a hypotonic solution because there's no solute in it. So a lot of your results will depend on how well hydrated your potatoes are to begin with. But typically if you put the potato cores in the hypotonic solution, the distilled water, what happens to them? So if we put this thing in distilled water, we had 3 of them so that we could have 3 repeats. Well, first of all, what did we do to measure the change, to see if there was a change? We measured them before and after, right? So they're all cut to 6 centimeters, and then we saw if they grew longer or shrank, right? So what happened after this thing was put in the distilled water? It grew, it got longer, right? Because it took on more water. And then you put the one in the 5 grams of salt solution, and what happened to it? It shrank because water went out. And the big thing is that the way they did the experiment, they cut them all the same way. Now, I've actually done this experiment in lab and had sometimes the lengths not change because it depends on how you bore the potato, right? You can bore it this way, you can bore it lengthwise. We come to find out that if you're paying attention, sometimes they don't grow in length. How else can they grow? In diameter, yes, they can get bigger this way. And so, we learned after a couple of iterations of this experiment that you really got to measure them in both directions to see if they change in one direction or the other. But one of the telltale signs is if they've gotten bigger, they also get a lot stiffer. We call this turgidity. They're rigid. And if you've ever seen plants after a good rain, what are they doing? They're standing up and blooming and, like this, because they've got a lot of water in them. They've got turgor pressure. And if you have basically a sunny day and the plants don't get a lot of water, what do? They kind of wilt. And then they stand back up again after the next rain. Very similar to what's going on with the hypertonic solution with the potato cores. They will shrink, and not only will they shrink, but they'll get really bendy. We call this flaccid. They get flaccid and bendy. So they shrink and they get bendy. Whereas the ones that were in hypotonic solution, they grew and they got rigid. The other thing is somewhere along the way, there was a solution that was very close to isotonic. And you didn't see much change in the potato core. So that's another thing to be aware of with this experiment. Now, we can talk about one thing very quickly, and that is the fact that water, as we say, moves down its concentration gradient via osmosis. So going back to our little U-tube over here, with our little semi-permeable membrane, we call the pressure of water wanting to move from one side of this tube to the other, we call that the osmotic pressure. So if we have more solute on side B than we do on side A, then side A has what we call osmotic pressure, and that's going to drive the water into side B. And that's going to force side B water level to rise and the side A water level to decrease. However, if you don't want this to happen, you can apply a pressure. You can stopper this thing, put a stopper in it, and if the stopper's in tight enough, then you can prevent this movement. And this is called hydrostatic pressure. Hydro meaning water, static means stop or staying the same. So if we put a hydrostatic pressure equal to the osmotic pressure, then this process will stop, right? As I say, this would be the hydrostatic pressure. And this will become very important when we look at cellular processes, and especially certain processes in like the kidneys later on, and in blood flow. So hydrostatic pressure is a pressure that's exerted against the osmotic pressure, right? Now what happens if we don't put this cap in very tight and the hydrostatic pressure is less than the osmotic pressure? What's going to happen? The osmotic pressure is going to be enough to overcome this hydrostatic pressure, and you'll probably pop the cap off, right? Right? So that's just some terminology here. So as long as this thing is allowed freely to flow, the osmotic pressure that is having more water concentration on one side will drive it to the other side. All right. So then we had the dialysis tubing experiment. Who got a chance to watch that video? Anybody in here? Okay, so the whole point of dialysis tubing, anybody ever worked with it? Oh. Well, dialysis tubing, as you probably know, comes as this thin tape, right? Looks like tape. It's really a persnickety thing because it's really a tube, but you have to kind of get it to open up. And you know those grocery bags at the grocery store that you can't get to open up, or the garbage bags sometimes? They're about 10 times worse than that. So basically you soak them in water and start to do this with them until they start to open up. And then they become a tube. Well, the thing about dialysis tubing is it has holes in it or pores in it of specific sizes. And that can be specified by the manufacturer. But the dialysis tubing that we chose for this experiment, is big enough to let water through and certain small dyes through, but not larger molecules like you'd have in salt, starch, and sugar. So what we did is we took this dialysis tubing and we tied it off at one end. It looks kind of like a little candy wrapper. And we filled it with some kind of solution. And then we tied it off at the other end, and we used dental floss. And typically, so we made these nice little things that look like candy wrappers with this solution in them. And then we put it in a beaker of some kind of other solution. And remember, once again, that these pores are big enough for water and small dyes to get through. And the dye we used was called methylene blue. There are other small dyes that can get through. And basically, we put the dialysis tubing in the beaker. So we have created two compartments now. Depending on what we put in what, we created one hypertonic and one hypotonic compartment. So for our dialysis tubing up here, in some cases, we might have put in a starch solution or a sugar solution. And then we put it in distilled water, right? And if we put this into distilled water, then this is the hypertonic compartment, and this is the hypotonic compartment, right? Now it acts just like a cell. We can watch and see what happens over time. And so what we did was, once we created this thing, this little dialysis tube, we got the outside of it nice and dry so that we weren't making any extra or measuring things outside of the dialysis tube. So we got this thing, we blotted it dry once we filled it, and then we weighed it, right? Then we stuck it in the solution for an hour, right? And then we took it out and weighed it again. In the case where we had the starch solution, and I think Dr. McCann used starch in his, we can also use sugar, salt, just about any of those. His had the starch solution inside the bag for one of the experiments, and he put the bag in the beaker and then left it there for an hour. So after an hour, what happened to the bag when we weighed it again? It got heavier, exactly. It got heavier because there's more solute inside. It had the hypertonic solution inside, so water from the beaker went into the bag. Likewise, he did the reverse. He put the starch solution in the beaker, but left the bag just pure water. So we had the starch solution in the beaker, and I think he used a 40% starch solution. And then we put the water, the bag which is water, in the beaker. And so what happened after the hour? The bag weighed less because there's more water in the bag proportionally to the beaker. And so the water left the bag and went to the beaker, right? So it makes sense. All right, he did another cool experiment to show diffusion. And he used something called methylene blue dye. And since I'm using blue for water, I'll just use green for our dye. And the thing about this dialysis tubing, it is small enough for our dye, our methylene blue, to pass through. So little green dots are the methylene blue. So he put the methylene blue dye in the dialysis tubing and stuck it in the beaker. What happened to the beaker after the hour? What happened to the water in the beaker? It changed color to blue. And so we had a very rich color, blue color, in the dialysis tubing to start out with. Some of that dye diffused out into the beaker. If you left it long enough and let it diffuse long enough, then they'd be about equal in color on either side because the solution would be uniformly mixed. If you do the reverse and start off with the methylene blue dye in the beaker and not in the bag, and then you leave the bag in there for an hour, what happens? The bag turns blue, exactly. And so I like to make the analogy that you have a bison farm, right? And you got a fence all the way around that keeps the bison in the corral, right? But you got some dogs there too, and you got little holes or spaces in the fence that are big enough for the dogs to get through. They can run in and out all they want, right? But the bison can't. So it's just like that. Anybody who's ever been on a farm or has seen this, you might have, and I've been on farms where dogs can get in and out of the fence all day long. And, okay, they're not supposed to, but they do a lot of times. But the horses or the steer or the cattle or the bison or whatever can't. And so it's permeable to the dogs, but not to the big livestock. And that's just exactly what's going on with these barriers. It's permeable to water and the small dyes, but it's not permeable to the large solutes. Now we have a joke in chemistry. For those of you who have taken chemistry, who's taken chemistry? Anybody? Okay. So you know what happens, and you've probably all seen this in real life too, right? So if you put too much sugar in your tea or in your drink, what happens to it? It is nasty. It tastes way too sweet. That's true. But what happens when you put so much sugar in it that the solution can't hold all of it? It goes to the bottom and sits on the bottom. We call that the precipitate. So there's a joke in chemistry. If you're not part of the solution, you're part of the precipitate. If you're not part of the solution, you're part of the problem. Yeah. All right. So that was that experiment. Everybody understands the purpose of these experiments. So the main thing, the reason we're using the dialysis tubing is it acts just like a cell membrane. So that's what we're using it. We're using it to simulate a cell membrane. And so long as you know that whichever one of these has the higher concentration of solution, that's where the water is going to go. So if you're asked a question on a test, we could pick anything you wanted. We could put, let's say we put 40% glucose solution over here in the dialysis tubing and stuck it in the beaker. And the question is, which way would the water flow? Into the bag, because it has more solute in it. And a real easy way to remember it, water follows solute. Water goes to where there's a higher concentration of solute. So, they could mix this up in any number of ways. Even if they put a 10% sugar solution in the beaker, water would still go into the bag because you have a higher concentration in the bag. Now, the osmosis would be a little bit slower, because some of the things that determine how fast these things move is the concentration gradient. That is the difference between them, right? So if you have only a small concentration difference, it's not going to move as fast, right? If you have a large concentration difference, it's just like if you're riding the subway and not too many people are on it, you're not in a huge hurry to get off when the doors open, right? But if you're in a jam-packed subway with 100 people stuffed in the same car, everybody's just wanting to get out as soon as they can, right? So that is one of the determining factors of how quickly these motion of molecules occur, both in diffusion and osmosis. Another thing that determines if you heat it up. If you add more energy to the solution, you're going to make these random collisions happen more often, right? So those are some of the factors that affect this process. So if we did this experiment at a higher temperature, it would go a little bit faster, right? If we did it in the refrigerator, it would go a lot slower, right? All right, so those are some of the factors that we're dealing with this experiment. Now, there's one other thing I wanted to go over, and that is part of the chemical level of organization. So I'm going to do a little bit of review for the test, the lecture test, okay? So there was on your study guide, and I didn't really do a lecture for this part, so I want to address it now. I said know the functional groups in the table on page 45. Now, there are many functional groups, and these are where molecules link together. Does anybody ever remember Schoolhouse Rock? Remember conjunction, junction, what's your function? Hooking up clauses and phrases. Well, that's exactly what the functional groups do. They hook molecules together, and they provide a way for molecules to link. And we have three that we're going to study, one of which is called hydroxyl, because as we will see, it has an oxygen and a hydrogen. This is a hydroxyl group, and it's on some molecule over here. And I'm just going to put R for the rest of the molecule, right? So hydroxyl is one of our groups. We have another one called carboxyl, which you recognize immediately, a hydroxyl with a carbon and another oxygen on it. So we call this carboxyl. And we've seen these in the head of fatty acids, right? Then we have one called amino, right? And this is on one end of an amino acid. And of course, we have this on the other end of the amino acid. We probably remember that. And then we have one called a phosphate group. And the phosphate group is basically a phosphorus surrounded by a bunch of oxygens. And depending on, I'm not going to get too much into this, but depending on the pH of the solution, sometimes these will have hydrogen ions attached to it, sometimes not. So this is why we have phosphoric acid and so forth, because it can give up protons. So in any case, this is our phosphate group. All of these are where molecules will link to one another. And so as we probably remember from the lectures, we probably remember that we had, remember our triglycerides that we talked about? They're built around the glycerol molecule, right? So we have a glycerol molecule. And as you recall, the glycerol molecule has a hydroxyl on it, right? Remember? And this is one of our functional groups. It has 3 hydroxyls, as a matter of fact. This happens to be a sugar alcohol. It has a 3-carbon backbone. It has these 3 hydroxyl groups. So we know it can link with 3 other molecules, right? Then we have our fatty acid over here, and it has a carboxyl group on one end of it. And remember how we just abbreviate the fatty acid like this? And the zigzag line just stands for carbons with 2 hydrogens coming off each one, right? So this is our fatty acid. And if we want to link this fatty acid to this glycerol molecule, well, lo and behold, look what we got here. We got this and this. And if we take those out, then we can make water out of them. And then we can link these 2 things together. So we have linked the fatty acid and the glycerol molecule, and we get water out of this. So this is a synthesis reaction, right? Because we have synthesized 2 molecules. into one, bigger one. In this process, we got water out. So we have this synthesis reaction is sometimes called a condensation reaction, but more specifically, we call this dehydration synthesis because we took water out of the molecule to synthesize these two molecules into a larger one. Does that make sense? Now what happens if we want to reconstitute our fatty acid and our glycerol molecule? What can we do? Put the water back in, exactly. And that is called hydrolysis because we're now using water to split the molecules back up again. And so all of our synthesis reactions that we're going to study in here are going to be dehydration synthesis. All of our decomposition reactions, breaking large molecules into smaller molecules, are going to be of the hydrolysis type. And hydro means water and lyse means to break, right? Like lysosomes. So hydrolysis is where we break the molecule using water. Everybody got that? So just a quick reminder, let's just say we stick a fatty acid on here. What do we got? We got one fatty acid on a glycerol molecule. What do we call that? Anyone want to take a guess? You've probably heard the term before. Monoglyceride?
Is that just one?
Yeah, it's just one. Mono is one. What happens when we put another one on here? Now we got a diglyceride, yep. And sometimes you hear the term diacylglycerol or triacylglycerol or monoacylglycerol. It's the same thing. So you've got monoglyceride. It's just easier to say monoglyceride, right? Here we got this one. What happens if we put a third fatty acid on here? What do we got? Triglyceride. Now what happens if this has a double bond in it? Now this is an unsaturated one, right? Because we put a double bond in. And as you recall, the double bond makes it rigid, right? Otherwise they're kind of free to spin. All right, so this is a triglyceride or a triacylglycerol. This is how fats are stored in the body. What happens if instead of, and because I said this thing can flip around, if it's a single bonded carbon, this thing can just spin around. We'll put our other hydrogen over here, and we'll put our OH group over here. What happens if we put something with a phosphate group over here? Then what we got? I'll draw a molecule. So I'll draw, and I'll just pick some random molecule. Actually, this is not going to be a random molecule. It's going to be something like this. And then we got, it doesn't really matter. So here we have some molecule. It's got phosphate groups on it, right? One of these phosphate groups can link over here, right? And we could link this molecule over here, and we'll end up with something like this. And then we'll have the rest of the molecule. And I'm not going to get too picky about what the rest of the molecule is, just that we have the rest of the molecule. So this is a phospholipid, right? And this is what makes up the phospholipid bilayer of your plasma membrane. And so as remember, the phospholipid is a double layer. And the question is why? And the reason is, remember our polar covalent bonds, and that polar covalent bonds tend to like water, and non-polar covalent bonds, like what we see with our carbon and hydrogens over there, they don't like water. So that means the head of this thing likes water, but the tails don't. Tails really don't like water. It's just like putting oil in water, and they don't mix. So what happens? Well, when we create our cell membrane, we have a bunch of phospholipids. I'm just going to abbreviate the heads as these little balls, right? So we got the heads of the phospholipids and their tails. And so they're going to line up like this. So here we have our phospholipid heads, and here are our phospholipid tails. And then we're going to have the other side of the membrane have its head sticking out, and the tails are going to aggregate inside, right? And now the tails are together. And there's no water in there, right? It's just hydrophobic tails. And the hydrophilic heads, one of them is facing the cytosol, or the inside of the cell, which is mostly what? Water, right? Extracellular fluid is over here, and it's mostly what? Water. So our hydrophilic heads are facing the interior and the exterior of the cell, respectively, because that's where we find water, right? The tails have all aggregated inside, where they can be next to each other, and there's no water in here. This is why water can't just go through the phospholipid bilayer. It has to have a channel. And so there's some proteins interspersed through the cell membrane every so often for channels for water that we call aquaporins, or water pores, water channels, right? Does that make sense? All right, so that is basically sort of the review for not only this lab, but for the lecture test, and some of the kinds of things you might see on there pertaining to the chemistry part, right? So has everybody got their review sheets, their study guides? So if you stick to the study guide, that's a lot of what will be in the test.